Good morning, we are from the Faculty of Engineering of the Benemérita Autonomous University of Puebla. Our project is called Evaluation of Antimicrobial Textiles using metallic particles supported on aluminum and silicate. The speakers are Professor María Luisa Castellanos Cabrera, Dr. Miriam Saraí Cruz Leal and Dr. José Ángel Juárez Torres. As an introduction, we can say that textiles represent ideal environments for the proliferation of bacteria since being in direct contact with the humidity of the human body can cause different infections. Hence the importance of developing antimicrobial textiles which can be functionalized either with chemical substances or technologies designed to kill or inhibit the growth of microorganisms such as viruses, bacteria or fungi. This can also cause bad odors, stains or different diseases. The development of antimicrobial textiles or functionalized textiles must use different technologies such as coatings, immersion in chemical solution, among others. But materials based on metallic particles such as silver, copper, zinc or quaternary ammonium stand out. In this sense, our main objective is to evaluate the effect of silver particles supported on zeolites as a potential material to inhibit bacteria on textiles. To do this, we have set three specific objectives. The first is to determine what type of textile fabric and materials are optimal for this study. 2. To implement the textile functionalization method and the antimicrobial material. And finally to carry out tests on the textile before and after to compare the results of the antimicrobial effect. For the experimental stage, we have divided it into two phases. The first referring to the preparation of the silver composite supported on VX zeolite. For the first phase, we use VX zeolites. Zeolites are microporous aluminosilicate minerals that have the capacity for cation exchange. We want to take advantage of this property and exchange it with metallic particles, in this case silver, and promote inhibition on the textile. For this, we make different concentrations of silver on the zeolite and we have named the samples depending on the number of cations exchanged. V1 for the sample with the lowest quantity of silver cations and V2 for the samples with the highest silver content. The exchange process is very simple. Basically, the weights of these concentrations are made and a solution is made with silver nitrate, which is the precursor, and the zeolite of interest is left in agitation for at least 24 hours and then the material is rinsed by centrifuge. And then the activation is carried out by drying in a muffle at 100 degrees Celsius for 1 hour. This material is recovered and we move on to the second stage. Stage 2. Textiles functionalized with metallic particles or zeolites. In Stage 2, the selection of textile substrates was made. To perform the functionalization of the textile substrates, two 100% cotton taffeta fabric materials were selected. The first of these was a sterile gauze as a dressing to protect wounds, and the second textile material was a compression bandage, also known as an elastic bandage. Substrate functionalization process. A colloidal solution containing the nanostructures of the synthesis identified as V9 was prepared in 20 milliliters of deionized water. Then moderate stirring was carried out at 60 revolutions per minute for 10 minutes at room temperature to achieve a homogeneous and stable suspension before placing our textile substrates. Subsequently, 0.0393 grams of gauze will be added to our colloidal solution, which was stirred for 20 minutes to allow the deposition of the nanostructures on the textile fibers to be as uniform as possible. After that time, 1% of an aqueous dispersion of nonionic acrylic copolymer was added to the solution, whose function is to generate a binder between the nanostructures and the fibers, Moderate stirring was continued for 10 more minutes until the wet process was finished. Immediately after which the textile substrate was placed in a radiation drying oven at an average temperature of 90 degrees Celsius for 60 minutes, this to achieve the curing of the acrylic copolymer on the textile substrate. Once the synthesis of the metallic particles has been carried out and the functionalization of the textile materials has been done, we proceeded to check the process of studying and understanding the properties that are sought to be achieved for this project, which is the preparation of antimicrobial textiles using metallic particles. Through scanning electron microscopy (SEM), we present to you the images of the textile substrate containing metallic particles supported on zeolites on the surface. In the micrograph identified with section A, we can observe the aggregates on the surface of the fibers, which is an indication of the existence of particles that are now part of the substrate. Likewise, what was observed in the micrographs identified with section B corroborates that there is the position of the particles in another region of the textile substrate. In short, the morphology of the fabric through the micrographs allows us to infer that particles of the synthesis identified as VX9 could have been placed. Conclusions. The functionalized material will be subjected to antimicrobial tests by cultivation of E. coli bacteria to analyze the efficiency as an antibacterial material. To know the degree of effectiveness, a culture of the bacteria will be carried out and the presence of the bacteria will be quantified. We can summarize that it was possible to support silver particles in the VX zeolite, both for low and high concentrations using the cation exchange method, and coatings based on particles called PMZ were also deposited, which is the acronym for metallic particles supported on zeolites, in this case on cotton fabric fibers using the immersion coating method. Likewise, through the characterization of the textile substrates by scanning electron microscopy (SAM), the presence and anchoring of the metallic particles is denoted, indicating that the method of immersion of the textile substrate in the colloidal solution is viable. Likewise, through the tests carried out initially in this case for the functionalization of the textile substrates, it was found that the method of deposition and anchoring of the particles can be scaled with textiles in real sizes as used in medical processes. We present to you the bibliographical references that have been building our research throughout this process. We thank you very much for the attention paid to the presentation of this project.